everyone, this is Tanima. This video is streaming in my YouTube handle at the rate just Tanima_Barbie. Just to let you know, my YouTube venture is totally a personal endeavor as a voluntary virtual social outreach, and it has nothing to do with my profession, where I'm engaged with research and teaching in the field of molecular biology and biotechnology in an institution in India. I personally believe that education should be freely accessible to all. And with this in mind, I have initiated a series on cancer biology. This is my sixth video on cancer biology. In continuation to my previous video, the present video discusses few more perspectives of unlocking phenotypic plasticity in the category of transdifferentiation in the topic of hallmarks of cancer. This video partially discusses the paper entitled Hallmarks of Cancer New Dimensions. The DOI of the same has been pasted in the description box. I would request you to turn on the subtitle or caption button on your screen in case you find difficulty with my deliberation. Do keep viewing. Members of the SOX family of chromatin-associated regulatory factors play a key role in determining cell fate and lineage switching during development. They are also linked to various tumor associated behaviors. A notable example of SOX driven transdifferentiation is seen in the development of therapeutic resistance in prostate cancer. In this case, the loss of RB and P53 tumor suppressors, which is common in neuroendocrine tumors, occurs in response to anti-androgen therapy but isn't sufficient on its own to trigger the conversion of well differentiated prostate cancer cells into neuroendocrine like carcinoma cells. These cells, which do not express the androgen receptor, exhibit molecular and histologic features of neuroendocrine cells. For this resistance to develop, there must also be an increased expression of the SOX2 gene, which plays a crucial role in driving the transdifferentiation of therapy responsive adenocarcinoma cells into neuroendocrine cell derivatives that are resistant to treatment. Another example illustrates how carcinoma cells use transdifferentiation as a survival strategy to evade elimination by lineage specific therapies. In this case, basal cell carcinomas or BCC of the skin treated with a pharmacologic inhibitor targeting the hedgehog smoothened or HHSMO oncogenic signaling pathway, which normally drives their growth, manage to resist the drug. The cancer cells achieve this by undergoing broad epigenetic changes in certain chromatin domains, altering the accessibility of two super enhancers. This results in the cells transitioning to a developmentally related but distinct cell type, allowing them to activate the WNT oncogenic signaling pathway instead. This new phenotypic state grants the BCC cells independence from the suppressed HHSMO pathway. As a result of this transdifferentiation, the cancer cell's transcriptome shifts from a gene signature characteristic of their original cell type, that is, the stem cells of the hair follicle bulge, to one resembling the basal stem cells found in the interfollicular epidermis. This form of transdifferentiation to achieve drug resistance is increasingly being observed in various cancers. Developmental lineage plasticity is also common among the various subtypes of lung carcinomas, including neuroendocrine carcinomas like small cell lung cancer or SCLC and non small cell lung cancer or NSCLC, which encompasses adenocarcinomas and squamous cell carcinomas. Single cell RNA sequencing have uncovered highly dynamic and diverse interconversions among these subtypes, along with distinct variations during different stages of tumor development, malignant progression, and responses to therapy. These findings reveal a more complex scenario than the simple idea of a clonal switch from one lineage to another. Instead, there are dynamically interconverting subpopulations of cancer cells that display features of multiple developmental lineages and stages of differentiation, complicating the approach to lineage based therapies for lung cancer. Researchers are now beginning to identify the regulatory factors that control this dynamic phenotypic plasticity. So, viewers, hope this video could provide further insights into the topic of hallmarks of cancer. Please don't forget to like and share the video and also subscribe to my channel. Here, I would like to mention that the references that were used have been pasted in the description box, and if you're interested to look into the details, you might check the same. Hope to meet you soon with yet another video on cancer biology. Till then, do take care, keep well, and bye.